Welcome to this explainer. Today we're diving headfirst into one of the absolute greatest medical detective stories of our modern era. We're going to walk through a decades-long hunt that culminated in the 2020 Nobel Prize in Physiology or Medicine. It is an amazing story of perseverance, brilliant methodology, and three scientists whose collective work literally changed the world and saved millions of lives.
Right, let's set the scene by traveling back to the 1970s. Picture going into a hospital for a routine, life-saving procedure, something like a blood transfusion. You expect to get better, naturally. But back then, receiving blood was essentially a terrifying game of Russian roulette. You see, a massive number of patients were walking out of the hospital with their initial problem solved, only to be infected with a severe, chronic, and completely invisible enemy. They were contracting hepatitis, but from an entirely unknown source. So the question was, how do you fight, let alone cure, a pathogen that you can't even identify?
1 million. Take a second and let that number sink in. That's the staggering number of deaths caused worldwide every single year by blood-borne hepatitis. It's just a massive astronomical figure, right? This made the disease a global health concern on a scale rivaling other historical heavyweights like HIV and tuberculosis. The stakes for solving this medical cold case absolutely could not have been higher.
Okay, Chapter 1. The Silent Global Threat.
By the 1940s, we knew there were basically two main types of infectious hepatitis. On one hand, you've got hepatitis A, which is transmitted through polluted water or food. It's acute, meaning it hits fast, but it generally has very little long-term impact. But on the other hand, we had a much darker picture, hepatitis B and what would eventually become known as hepatitis C. These are transmitted through blood and bodily fluids, and they aren't short-term at all, they're incredibly insidious and chronic. Categorizing these differences was crucial because it successfully narrowed our suspect pool down to the much more dangerous, blood-transmitted varieties.
And as you can see, this isn't a disease that strikes you down overnight. It is terrifyingly silent. An otherwise completely healthy person could be infected, and as the virus quietly damages their liver over 10 to 30 years, it progresses from a healthy liver to chronic hepatitis, then to cirrhosis, and finally to hepatocellular carcinoma, which is liver cancer. The patient wouldn't even know they were sick until serious, often fatal complications popped up decades later.
Which brings us to Chapter 2, a mysterious missing virus. Enter our first medical detective, Harvey J. who was working at the U.S. National Institutes of Health. Now, back in the 1960s, a scientist named Baruch Blumberg had actually discovered the hepatitis B virus, which earned him a Nobel Prize. Blood tests for Hep B were quickly created. But Alter noticed something deeply troubling. Even after screening donated blood for hepatitis B and completely ruling out hepatitis A, a huge number of patients were still getting severely sick after transfusions. Just listen to the stark reality of the time in this quote. A significant number of those receiving blood transfusions developed chronic hepatitis due to an unknown infectious agent. Think about the sheer panic of that realization in the medical community. They were doing everything right, testing for everything they knew about, but this ghost was slipping right through the cracks. It meant the blood supply was profoundly unsafe, and they had absolutely no idea what was causing it.
But here's the crucial point. Alter didn't just throw his hands up and quit. He methodically proved that blood from these sick patients could actually transmit the disease to chimpanzees. He showed that this infectious agent had the specific characteristics of a virus. By ruling out all the knowns, his meticulous work to find the parameters of this missing suspect, the scientific community was left with a terrifying new placeholder name for the disease, non-A, non-B hepatitis.
Let's move on to Chapter 3, Hunting the Genetic Code.
Meet our second medical detective, Michael Houghton. Working for the pharmaceutical firm Chiron, and after over a decade of incredibly frustrating dead ends, Houghton realized he had to take on a seemingly impossible, totally untested task. He had to hunt down the invisible virus's genetic sequence.
Okay, let's break down Houghton's strategy here because it is basically a masterclass in lateral thinking. It was the ultimate needle in a haystack problem. Step 1. He extracted a massive collection of DNA fragments from the blood of an infected chimpanzee. Step 2. He made this incredibly bold assumption that somewhere, hidden among millions of the chimp's own normal genetic fragments, there would be tiny pieces of the unknown virus. Step 3 is where the real genius lies. He took blood serum from human hepatitis patients, blood that should contain natural antibodies fighting the disease, and he used those human antibodies to scan the chimpanzee DNA fragments. The logic was brilliant. If the human antibody reacts to a fragment, that fragment belongs to the virus. And step 4, after a truly comprehensive, exhausting search, they found it. One single positive clone.
Boom! That single positive clone blew the case wide open. Further analysis revealed that this tiny genetic fragment was derived from a completely novel RNA virus belonging to the flavivirus family. The presence of those human antibodies firmly implicated this newly discovered agent as the culprit. Finally, the ghost had a name: Hepatitis C. This was an absolute, undisputed milestone in medical history.
Chapter 4, the final missing piece. This brings us to our third laureate, Charles M. Rice, a researcher at Washington University in St. Louis. Now, Houghton had found the virus, sure, but Rice knew that the case wouldn't truly be closed until one incredibly specific standard was met.
The ultimate question he had to ask was, could this newly cloned virus alone actually cause hepatitis? Was it the sole murderer, or was it just an innocent bystander found at the scene of the crime? To definitively close the case, scientists had to prove that the cloned virus was able to replicate itself and directly cause the disease without any other factors present at all. Let's walk through exactly how Rice brilliantly solved this. He noticed a previously uncharacterized region at the end of the hepatitis C genome that he suspected was the actual engine for virus replication. He also noticed that the isolated virus samples had genetic mutations that were hindering it from reproducing in the lab. So using advanced genetic engineering, Rice essentially cleaned up the code. He generated a pristine RNA variant of the virus completely devoid of those inactivating mutations. When he injected this purified, engineered RNA into the livers of chimpanzees, the results were undeniable. The virus replicated, it was detected in their blood, and it caused the exact same pathological changes seen in humans with the chronic disease. This was the smoking gun, folks. It was the final, undeniable proof that hepatitis C alone caused the unexplained cases of transfusion-mediated hepatitis.
And finally, Chapter 5, Curing the Incurable. Just look at the massive domino effect of these breakthroughs. Because Alter defined the disease, Houghton sequenced it, and Rice proved how it worked, highly sensitive blood tests were rapidly created. Almost overnight, post-transfusion hepatitis was essentially eliminated in many parts of the world. The global blood supply was finally safe. But they didn't stop there. Understanding the virus's engine allowed for the rapid development of targeted antiviral drugs. And today, for the first time in human history, this previously invisible, fatal disease can actually be completely cured.
So, the 2020 Nobel Prize cemented this as a truly monumental achievement. Today, we have the incredible physical manifestations of their work, highly sensitive tests, and effective antiviral pills. But as you look at the global landscape, it reveals a pretty stark truth. A golden breakthrough in a lab doesn't instantly erase global inequities. The disease still ravages populations who just lack access to these life-saving resources.
So, I want to leave you with this final provocative thought. The scientific mystery is solved. The medical detectives have done their job and handed us the ultimate weapon. With the power to cure hepatitis C finally in our hands, how quickly can we overcome our global inequities to completely eradicate this disease from the face of the earth? The science is ready, now it's really up to us. Thank you so much for joining me for this explainer. Keep asking those big questions, and I'll catch you in the next one.